All right, hey everybody. So as we learned last week, these cells in our retina analyze our visual world as an array of spots of light, and then information about the spots of light that's hitting the eye is sent along the optic nerve to the brain. So today we're going to see how the visual cortex, or the striate cortex, our primary visual cortex in the occipital lobe, really receives this information and reanalyzes the visual world in terms of bars and edges of light, basically translating it from spots into stripes. So this is the path of image processing, basically what path the information takes from the eye to the striate cortex at the back of the brain here. So from the eye you have, or in the eye, in the retina, you have the vertical path from the photoreceptors to the bipolar cells and then to the retinal ganglion cells. The axons of the retinal ganglion cells form the optic nerve. And remember, in the eye, there's also lateral connections between, by using horizontal cells between the photoreceptors and bipolar cells. Remember, they perform lateral inhibition, so creating that receptive field and borders and edges. The other lateral connections are made by amacrine cells, which synapse between bipolar cells and retinal ganglion cells. Right, so amacrine cells are involved in contrast processing and measuring timing of visual inputs and lots of other functions that aren't too well understood yet. So the retinal ganglion cells, as I said, the axons form the optic nerves. The cells are going to fire action potentials, and the information is going to come out and possibly cross at the optic chiasm here. We'll talk about that more in detail later. And then it's going to go to the nucleus in the thalamus that's important for visual processing, called the lateral geniculate nucleus. And then it'll go onward to through these optic radiations to the primary visual cortex. It's called striate cortex because it looks stripy when it's stained and examined under a microscope. So we'll see more about that in a bit. This is just showing you the occipital lobe at the back of the brain, and that's where our primary visual cortex is. And then some other association areas where some higher order visual processing occurs of more information. So yeah, the occipital lobe, basically the furthest away you can get from the eyes. I'm not sure why it developed like that, but there we have it. So by the time we're done, you're going to know all of this. But for now, I just showed it to you to kind of scare you a bit, or maybe excite you about what you're about to learn. So vision scientists basically have a bunch of ways to test visual acuity. Acuity is the smallest detail that you can resolve. So some people will have better visual acuity than others. And so for this test, what we'd do is we'd see how far away you'd have to stand for you to not be able to tell the orientation difference between the two sets of these stripes. Right? So you can obviously see that one set's vertical, one set's horizontal. And so the idea is that you at some point when you're far enough away, both patterns are just going to look like gray blobs, and you won't be able to detect which orientation is which. Another way to do it would to be having you stand at a certain distance and then varying the size of the gratings, and you're trying to distinguish them. That's actually the more common method since it's easier to control. You don't have people like walking around and moving back and forth. So yeah, usually that's what they'll do in lab studies. And so eye doctors generally use the distance that you would stand away to characterize your acuity. So I'm sure you've heard of 20/20 vision. Basically, the number / the number is your distance that you can see versus the normal vision distance that someone else would be able to see, the average person. So then, is 20/40 vision good or bad? It's bad, right? So the idea with 20/40 is that you can see at 20 feet what the average person can see at 40 feet. You'd have to be twice as close, right? So like 20/15 or 20/10 vision, that's good. You can see at 20 feet what a normal person could see at 15 feet or 10 feet. Right? So that means you could stand farther away from the target than normal and still be able to make it out. So the 2020 method for characterizing visual acuity is okay, but it's not as precise as it could be since it does rely on a specified target size and also specified distance. So vision scientists instead will characterize acuity in terms of visual angle that's required. So using the visual angle, it automatically incorporates size and distance. So what they'll do is they'll record the smallest visual angle where you can actually perceive one cycle of the grading. So black or white or black or yellow in this picture. Basically, a cycle is a change from dark to light in a repeating pattern. So basically, the smaller the visual angle is, where you can identify a cycle of a grading, then the better your vision is. And remember, the further away you get from something, the smaller the angle will be of the information coming into your eye. Or looking at another way, from a fixed distance, and the smaller the object is, the smaller the visual angle would be. So that's how the two measures of acuity relate to each other. In terms of spatial frequency, that's the cycles of grading per unit in a visual angle, so measured in degrees. So another way to think of that is the number of times a pattern repeats per unit area that you're measuring. So in this figure here, you can see that there's very low spatial frequency in A, B is a bit higher, so medium spatial frequency, and C has a high spatial frequency. Another measure is contrast sensitivity, which is your ability to be able to figure out that there's a difference in brightness of areas side by side. So definitely it's related to spatial frequency, right, as you can see. But the idea is, can you see a difference in these two side-by-side areas? So a couple of factors that influence contrast sensitivity are shown here. The first one in A shows the idea that as the lighting conditions, photopics, bright light, scotopics, dim light, mesopic is like average light. So our contrast sensitivity decreases as it gets darker and darker. It makes sense. And then C here is showing you age. So you can see that when we start out, our contrast sensitivity is okay, it gets better, and then as we age, it gets worse and worse and worse. Right? So it does tend to decline with age. So our visual system is very smart. It's going to break down images into a bunch of components. And each of those components is going to be one of these repeating waves called a sine wave that has its grading with a particular spatial frequency. So our brain's ability to do this is called Fourier analysis. So that's also how our perceptual system deals with things like sound waves. So even something as artificial as like a square wave, right, that could be broken down into gradings, like black and white gradings. And so it can be created by summing together a bunch of different sine waves of different frequencies. Now getting back to those center surround receptive fields, how do those respond to sine wave patterns? So if you look at the curve with the pluses and minuses here, it shows how, in the middle of a sine wave grading with, the brightest intensity, you have the plus, and the sides, minuses, right? So the idea is that, rather than circles, this pattern represents a slice through a circular receptive field. And the plus portion represents the on center, and the minus portion represents the off surrounds. So in A here, when the receptive field sits on a grating that has low spatial frequency, the on center portion is in a bar of light, but so are the off center or off surround portions. So the cell is going to have this excitation pattern that you see here. It's going to be excited by light falling on its center, but it's going to be inhibited by light falling on the surround. In B here, you can see this is a good situation, so the spatial frequency of that sine wave grading fits pretty perfectly with the retinal ganglion cell's receptive field. So the on-center portion of the cell sits within a light bar, and the off-center portion sits around the side within 2 dark bars. And then here you can, so then it would have this great firing pattern, like high firing frequency. And then in C here, you can see that the sine wave pattern has too high of a spatial frequency to excite the receptive field optimally. So the on center gets both light and dark bars, and the off center, off surround does too. So none of the portions in the receptive field are getting their optimal stimulus. So then the corresponding response from the neurons is pretty weak. It's not firing a lot of action potentials. Oops. So spatial frequency is definitely important, but also the phase is important, which is the position within a receptive field. Right? So here in A, you're seeing the sine wave grating that's in a perfect phase to maximally excite the cell. You can see the on center is in line with the white. In B, there's portions of the on center receptive field that are covered by a dark spot, and portions of the off surround as well touch a light spot. So it's not going to respond maximally. In C, the cell gives a negative response because the input that it's receiving, its on center, is actually in the dark spot, right? So it's receiving the exact opposite of what it wants. And then in D, there's not going to be a big response, or maybe none at all, because the same reasons as in part B. So we have 2 lateral geniculate nuclei, which one on each side of our brain. And that's where the axons from the retinal ganglion cells are going to synapse. So this image shows a cross section of a lateral geniculate nucleus from an ape, and it's artificially colored, to show the ipsilateral, so same side projections in blue, and the contralateral, so opposite side projections in yellow. So in those connection layers, they're not going to look like that in your brain. But the idea is that, and you can also see further division. So the inner 2 layers, 1 and 2, have magnocellular, so bigger cells. Those cells, remember, respond best to motion stimuli. And then the outer 4 layers, 3, 4, 5, and 6, they're parvocellular cells, which respond best to color and shape stimuli. So notice that the LGN, it's going to have some ipsilateral connections and some contralateral connections. And the organizing principle is that the right LGN processes information from the left visual field, and the left LGN from the right visual field. Also, the LGN has an very ordered mapping of the world, which is known as topographic mapping. So I mentioned this, right? The types of cells in the LGN. We've got the magnocellular ones with they're large. The bottom 2 layers, or the inner 2 layers, are them. You can see on the figure that they're bigger. They receive input from the magnocellular retinal ganglion cells and respond to movement and moving objects. The parvocellular cells, they're smaller. They're the top 4 layers. Receive input from parvocellular ganglion cells and respond best to really fine spatial details and also stationary objects that aren't on the move. And then the coniocellular cells are really small. They're between the magnocellular and parvocellular sections. So if you got really deep in there, you could see them. And we're still not entirely sure what these cells do. So most of our early information about cortical layout came from animals and humans that had cortical lesions. So what we know is kind of shown in A here, but from those studies. But now we use MRI and fMRI, and we get a lot clearer results, because MRI, remember, reveals the structure of the brain, and then fMRI reveals brain activity through looking at blood oxygen, so using that bold signal. And results from the fMRI studies are shown here in B. Right? So With our striate cortex, our primary visual cortex, there's about 200 million cells, and there's a major transformation of visual information that takes place there in the striate cortex. For example, there's circular receptive fields that are found there in the striate cortex. That are found in the LGN. They end up getting replaced with elongated, stripy receptive fields in the cortex, which of course helps give it its name. So cells in the striate cortex, they're going to respond best to what? To bars of light, right? Rather than spots on your eye. This shows the cellular layers of the striate cortex. So each of the layers serves a different function. And the input, output, and processing at the end actually is represented in each of those different layers. So yeah, you can see kind of the laminae or layers, right? Different from the columns that go up and down. Right? So they're each going to serve some kind of different function. So there are a few important points to note about the striate cortex. It has topographical mapping, right? So in terms of where the image hits the retina, it's going to hit roughly the same spot in the cortex. Also, there's cortical magnification, which means that there's a lot of scaling of information from different parts of the visual field. And there's a lot more cortex devoted to processing the information from your fovea, which is important for visual detail, than cortical space dedicated to processing the periphery. Right? So think about the right side of your eyeball, and it's showing you a pretty good figure here. Think about the right side of your eyeball, and it's going to see the left side of the world, right? And vice versa. The left side will see the right side. The right side is processed again by the left hemisphere, the left by the right. Another point is that one half of each eye, as you can see, has a same side or ipsilateral connection to the striate cortex, and one half of each eye has the contralateral connection. Right? So then the idea is that the left visual field goes entirely to the right cortex and right visual field to the left at the end. So the central part of the visual field that's near the fovea, it gets overrepresented, it gets magnified, right, in the cortex. So you can see kind of in the striate cortex, there's the woman's face is distorted and enlarged due to this cortical magnification. In terms of moving away from the fovea, so that's eccentricity, how far you get from the fovea, your ability to see the fine details, so your acuity decreases the further you get. I'm not sure why this got cut off like this, but the idea is that cortical magnification is great to help you see the fine detail, but any images that are in your periphery, they have a lot lower resolution than any images at fixation. So this figure is trying to show you that. So if you fixate the E at the left from the correct distance, then each E on the right of it will be easy enough to discriminate. But yeah, so the closer something is to your fovea, then the easier it is to see, and so the smaller it can be. This is due to our high spatial acuity cones that are in the fovea and the extra processing of the fovea in the striate cortex. But if you focus on a big E, then the smaller ones that are in your periphery are not going to be. You're not going to be able to figure them out. You're not going to be able to see them. So this idea, this fact, can lead to visual crowding, which is the effect of clutter on your peripheral object detection. So basically there's just too much stuff, and then it has a negative effect on your ability to detect objects in your periphery. So stimuli that could have been seen if they were in isolation in your peripheral vision, they are now really hard to discern when it's cluttered, when there's lots of stuff. So, this is a bottleneck for visual processing. Right? So if you fixate on an image, on this image, if you fixate on the circle in the center of the image, then that makes a small child, a lot harder to see that's closer to you. So, fixating on it, and if you compare the two kids who are like equally distant from the circle, the one on the left is a lot harder to see than the one on the right, right? Because of the clutter, the signs and stuff around the child. If you fixate on, other stuff, you'll see the same thing is true. So say in B here, if you fixate on the plus signs, the shapes along the top of the image are much easier to discern and to see boundaries and stuff. So your acuity is much better than the stuff on the bottom, right? So definitely the more distractors there are, the more clutter there is, then the harder it is for you to make out those fine details. Now remember that cells in the striate cortex respond best to bars of light. So they are tuned to bars that are particular orientations. So certain neurons in the striate cortex are going to respond most to bars that are at the orientation that they're tuned to. And then the response rate gets worse and worse when there's a big difference from the preferred orientation. So the figure shows that here. So the firing rate of this neuron in the striate cortex, it responds best to a bar that's vertically oriented, right? And perfectly vertical, not on an angle at all. If the bar is tilted to the left or the right on like a 45-degree angle, then the cell still fires a little, but at a very, very reduced rate. Now, the more vertical you get, the better the cell fires. And then if the bar is horizontal, the cell doesn't respond at all. Right? So this is true of most neurons, maybe all neurons in the striate cortex. So you might be wondering how those circular receptive fields that we saw in the LGN that get transformed into this long bar receptive field in the striate cortex. And it seems Hubel and Wiesel found that a cortical neuron that responds to bars, that they receive input from several different retinal ganglion cells. And if you string those retinal ganglion cells all together, they actually form a bar. So their receptive field is like a bar now. So now a cell that's tuned to any orientation you want could actually be created by connecting it up with other retinal ganglion cells that respond along this certain orientation of a bar of light. And it's not just bars of light that they respond to. There's lots of different cortical cells that respond well to movement, so moving lines, bars we just talked about, edges, and gratings of certain frequencies or possibly certain orientations, certain motion directions if they're motion responsive. And so they respond to these very specific characteristics. And since they do so, they actually function like a filter for filtering that portion of an image that excites that individual cell and not responding to kind of the rest of them. In terms of the bars of light, some neurons prefer certain bars of light that are of certain lengths. So this is called end stopping. So basically the figure is showing that this particular neuron has this receptive field that it's going to respond best, so generate the most action potentials, when the bar of light fits exactly in its receptive field. It'll respond less when the bar of light is either too long or too short. So there are lots of complicated receptive fields definitely in the striate cortex. And each of these cells can respond to input from both eyes, but the idea is that they might respond preferentially, and cortical neurons tend to have a preferred eye that they respond to, so they respond more vigorously, so more action potentials to the input from one eye or the other. But the input has been combined from both eyes when it's got to the primary visual cortex. So also, the neurons can adapt to continued presence of a stimulus. So adaptation is a reduction in response caused by prior stimulation or continuous stimulation. And we'll see adaptation in every sensory system that we look at. So if you present a visual stimulus for an extended period of time, the brain basically says, I've seen this before and I'm bored, and it adapts to it. It stops responding. And this can actually be exploited to kind of knock out groups of neurons by exposing them to a certain stimulus or a certain type of stimulus, and then they'll stop responding for a short period of time. And so the figure shows what happens here. So there's this response pattern for this particular cell. So it prefers these vertically oriented bars. And then if you expose it to a certain orientation of light, so this bar that's oriented at 20 degrees, if you expose it to that input for quite a long period of time, then eventually the cell becomes kind of accustomed to this input and just doesn't respond to it as much anymore. And you can see that after this prolonged exposure, it's not responding very well to it. So the cell's orientation tuning has been affected so that it has a higher rate of firing for stimuli now that are slightly leftward tilted. So it's going to adapt in kind of both directions. So this demonstration actually allows you to experience selective adaptation for yourself. So what you're going to do is you're going to move your eyes left and right along this dark black fixation bar in the middle of A. Do it for like 30 seconds. And then when I tell you to, quickly look at this fixation point on the right. Actually, just do it yourself for 30 seconds, 30 to 60 seconds. And then when you're done that, look at this fixation point on the right. So if you do this, hopefully you did, do you notice anything different about the top and the bottom gratings? Right? So the top grating should now appear to be tilted slightly to the right and the bottom one to the left. So what you've just experienced is called the tilt after effect, and it's due to adaptation. So it's a perceptual illusion because your visual cortex has adapted to a pattern in a certain orientation. And this supports the idea that our visual systems contain different neurons, like individual neurons that are selective for different orientations, and that they can adapt after repeated exposure to a certain stimulus. Selective adaptation like this can also occur for spatial frequencies. So again, this is evidence that our visual system contains neurons that are selective for certain spatial frequencies. So basically you can do this again. So you start by staring at this vertical grating for 30 to 60 seconds. And then you look at part B, right? So then rather than the inverted U shape that it normally has, it should have a profile similar to the red line that you see in C. So this notch in the curve, rather than being able to see like all of the gratings, you have a notch in where you don't see them very well. That shows the reduced sensitivity that resulted from staring at this vertical grating. So you had selective adaptation. And so what effect would it then have if you did this with this image in part D here, right? In terms of looking at this in B. And that's going to be your participation question. Okay, so definitely submit your answers online. within the next 3 days. Okay. And I will make sure that the answer is there for you as well. So these types of experiments, adaptation experiments, they do provide some good evidence that orientation, both orientation and spatial frequency, are coded separately, as I mentioned, by separate neurons in our visual system. So some research has told us that our vision is coded in spatial frequency channels, which are basically pattern analyzers. They are created by a whole ensemble of cortical neurons, and each set of these neurons in the ensemble is tuned to a limited range, a certain range of spatial frequencies. So these different spatial frequencies are going to emphasize different types of information in our visual system. So just look for a sec at this complete image in A, and then you can see reconstructions of it. So if you remove low spatial frequencies, you can see what you'd see in B. So only high spatial frequencies. And then if you remove high spatial frequencies in C, then it looks quite blurry. You only have the low spatial frequency image. So basically the idea is that you have these ensembles of neurons that kind of compute all of that information together. So who's hiding here? Right? So it's hard to tell who this famous person is, but if you just try squinting, then that squinting removes your high spatial frequency information, and it kind of reveals the low spatial frequency energy that's present in the image. So the low frequencies enough alone should be able to let you recognize that it's Abe Lincoln. So yeah, the idea is that there are these certain channels that help you recognize high, medium, and low spatial frequencies, and everything in between as well. In terms of developing our spatial vision, usually infants tend to look at more complex stimuli. They don't just want to look at kind of a circle. They want to look at a circle with gratings in it. So this can be studied by using the forced choice preferential looking paradigm and visual evoked potentials, so using EEG while they're looking. So this is definitely a behavioral response that we can measure experimentally. So these visual evoked potentials are the electrical signals from the brain. They're evoked by looking at visual stimuli, and then you measure kind of the response in the brain while a baby or a person is looking at visual stimuli. So if there's a difference in the amount of time that the baby looks at two different stimuli, then that means that they can tell the difference between them and that they find one of the two more complex or more interesting or more surprising. We don't know exactly why they look at it more, but it seems they like it more for some reason. So as you can see here in B, experimenters might use electrodes on the baby's head to measure the visual evoked potentials so that they can measure an infant's ability to sense differences in contrast. And they can do this over, many, many trials. So if the baby can see the image well, then the visual evoked potential should be a bit weaker. And then C here is showing some data that plots the visual evoked potential strength as a function of spatial frequency on the x-axis here. So you can see that the greatest visual evoked potential response came from low spatial frequencies, which is consistent with the fact that young babies can't really see high spatial frequencies that well. So they preferred to look at kind of the low spatial frequency images more. Generally, young children are not very sensitive to high spatial frequencies because their visual system is still developing and the cones and rods are still developing and even taking their final shape. Also, retinal ganglion cells are still migrating. They're still moving to their eventual destinations and making connections with the photoreceptors and bipolar cells in the fovea. The fovea itself isn't actually fully developed until like 4 years old. So it makes sense that the fine details, aren't seen very well until 4 years old. Okay, now this is just a little activity to do quickly. Look at the movie, the little movie playing here. And when you're ready to move on, I'll click. Good. Now click here. Now look at it again. Okay. Now look at it again. Now look at it. Ooh, tricky. Okay, so you just participated in an experiment that's usually done with babies. The procedure is called habituation. So the fact that they will be interested in something the first time they see it, and then if you encounter it over and over again, you just get bored with it, right? So the first time the babies, look at it, they look at it for a bit, and then they stop looking as they've seen it more and more, right? But now this time in this situation, you saw a different movie, right? So most people will look longer, including babies, at this movie than they did because, hey, something's different, right? So an increase in time that you spend looking at something is indicating that you can see a difference, and also that differences are interesting to you, right? So obviously you could tell someone if you see a difference, but 4-month-olds or 1-year-olds, they can't tell you what they see, right? So now you can use procedures like this. So pretty sweet, eh? This is just looking at like a case study, basically, of a child who had abnormal early visual experience because she had a severe cataract in her left eye. So basically, she only had one eye. So she had monocular vision instead of the regular binocular vision. And that can cause huge differences in the cortex in terms of the physiology of it, the functioning of it, the organization of it. And that results in devastating and permanent loss of spatial vision because you need both of your eyes. So amblyopia is a problem where there's not spatial vision in an otherwise healthy eye. Strabismus is also known as lazy eye, so a misalignment of the two eyes. And anisometropia is a condition where the two eyes have different refractive errors. So if a person has cataracts or strabismus, that can actually lead to some pretty serious problems. So detecting these things early and dealing with them can prevent those problems. But if they're not detected early enough, there seems to be a critical period of development in between 4 and 5 years of life where problems can be corrected. And if they are, then pretty normal vision will develop, if not completely normal vision. But if they're not corrected and input from one eye is limited for some reason, then the neurons that were meant to process that eye, they might get reassigned to other purposes, and they might get reassigned to process the other eye, for example. So now, later on in life, they don't have the proper neural functioning for that eye. So even if the problem is corrected later, then it's kind of too late because they've passed that critical period and there's been a lot of reorganization. But again, if it's corrected early enough, then the process can be reversed. The next thing we're going to talk about is space perception and binocular vision. So basically, this is how the visual system accomplishes the task of perceiving our 3D world, but using 2D images that we form on the back of our retinas. So I think it should say depth perception instead of space perception, and you'll see why. But yeah, that's what it was called. Okay, so if you remember your Euclidean geometry, parallel lines remain parallel as they get extended in space, and objects remain the same size and shape as they move around in space. And also, internal angles of a triangle always add up to 180 degrees, and so on. So now, from what we know about the retina and cortical magnification, we know our brain is not seeing images this way. Okay? Therefore, our brains work with non-Euclidean geometry all the time, even though we're not aware of it. Right? So there's not the normal rules in the brain. So the brain does a few things. So probability summation is our increased probability of detecting a stimulus because we have 2 or more samples. That's one advantage of at least having 2 eyes. that face forward and not, one eye in the front, one eye in the back, right? We have more than one image hitting our brain. And then binocular summation is combining the signals from each eye that makes performance better with using both eyes than just using one eye. If you think about it, the two retinal images of our 3D world, they're not exactly the same. They're slightly different. So your brain takes them and combines them in a way that makes you perform better than if you wore a patch, like a pirate or something that we talked about before. So I'm sure you already know, that it's better to have two eyes than one. But here's a way you can verify it for yourself that your two eyes see different things. So you take your two fingers, put them like so, and then close one eye and then close the other, and you see different things. Okay? So that is binocular disparity, the difference between the two retinal images of the same scene. So it's the basis for stereopsis, which is our vivid perception of the 3D of the world that we can't get with just using one eye binocular vision. So our brain uses lots of different cues. So a depth cue is information about the 3D world, so depth of our visual space. Monocular depth cue is available even if we only have one eye, and a binocular depth cue relies on information from both eyes together. So what you can see here is the different visual rabbits have. They have a very wide field of view, both in front and around them. And above them, too, because their eyes are kind of on top of their head. Very little, though, of their visual field is seen by both eyes, as you can see. So they have often to use mostly binocular depth cues. Humans have a narrower field of view, like we're not seeing behind us, but much of our visual field is seen by both eyes, which allows us to use binocular depth cues and use that information to help us navigate through the world. So this picture would probably melt a rabbit's brain, and maybe yours, because the depth relations in this picture don't add up. So since the 3D world is projected onto our 2D retinas and then our brain has to reconstruct that lost dimension of depth, our brain uses cues to determine depth, but it's of course not foolproof, which is what this picture demonstrates. So now let's see the cues our brain uses. So this is a monocular cue, occlusion. Basically, it's a cue to the relative depth order, but it doesn't tell you for sure. So, for example, if one object, like the circle, partially obstructs the square, which partially obstructs the triangle, there's three shapes here, and we would typically assume that they are at different depths, right? With the triangle being the furthest and the circle being the closest, and that's occlusion. But now you can see two possible interpretations of the situation. So sure, it's possible that the square and the triangle are actually the same distance away as the circle, but that they have pieces cut out of them. But yeah, the more likely possibility is that the three shapes are whole figures that are at different depths. So that's occlusion. Another cue we use is metrical depth cues. So based on these depth cues, you know exactly how far away something is. So this might be useful if you're trying to do stuff like catch a baseball or something like that without it hitting your head. So it gives quantitative, like numerical information about distance. So like 100 yards away, there's a yield sign. Right? I don't know why it says stop. Non-metrical depth cues are cues that do provide still information about depth order, but not the size. Right? So it's not giving you quantitative information. So it tells you which object is in front or behind other objects, but you don't necessarily know how far away the object is from you in a number sense. Relative size is another one. Basically, you compare the size between items without knowing the absolute size of either ones. So in this one, with all things being equal, we assume that smaller objects are further away from us than the larger objects. So these balls seem to be all at different depths because of this relative size cue. But again, it's possible that they're just different sized balls all sitting on a black table. Right? In reality, since this is a picture, The balls are, in fact, on the same surface and are different sizes. But most of us would interpret 3D structure here. And if you saw any 3D structure, it's because of this relative size cue. Relative height is for objects that touch on the ground. So those ones that are higher in the visual field are further away from us. In the sky, though, the opposite is true. So objects lower in the visual field appear to be farther away. So depending on the horizon, basically, you use this relative height cue. Texture gradient is another depth cue that's based on geometry. So that fact that items that are the same size, they form kind of smaller and then closer spaced images the further away they get. Right? So a texture gradient you can see here of rabbits. We're using rabbits a lot today. So the rabbits in the top here appear to be farther away than the rabbits at the bottom because of the cue of relative height and relative size, but they've also formed a texture gradient as well, right? So there's a combination of relative size and relative height, and that gives you texture. This is a different texture gradient of rabbits, right? So the rabbits in this image get smaller as you move from left to right, and the sense of depth stratification is actually weaker than in this image here. Right? And yet another texture gradient of rabbits. So check out the bunnies on the upper left and bottom right. Which one's bigger? Right? They're exactly the same size on the screen, yet the rabbit on the lower right looks smaller because the texture gradient makes it so that the rabbit in the upper left looks farther away. So it must be bigger to project that same visual angle on your retina as the rabbit on the lower right. There's also a size contrast in terms of the objects around it. So it makes them look smaller because of that size contrast. Another cue that you use to determine depth is familiar size. So we know the typical size of objects, right? And when you know that, the typical size, you can guess how far away something is based on how big or small it looks. So this often works in conjunction with the cue of relative size. So they work together. So in this picture, you can see the hand on the left, it looks closer than the hand on the right because it's huge. And we know how large hands generally are relative to the size of a woman's body. So the one on the left here, you're definitely like, yeah, it's closer to me than the hand on the right. So definitely relative size and relative height gives some metrical information to us. And a relative metrical depth cue is one of those cues that could tell us that object A is twice as far away as object B, but it doesn't give us information about absolute distance to either of them. So it's relative to the other objects that you're looking at. So if you imagine that all three of these spheres are touching the ground, then the green one's furthest away. If you imagine that they're all floating, then the green one is the same distance as the blue and red ones, but it appears to be smaller. And so it gives you some relative information about the depth of that. Familiar size also provides some precise metrical information as long as you know the actual size of the object and the visual angle it usually takes up on your retina. So it can give you that information about how close or far something is. Absolute metrical depth cues provide quantifiable information about distance, of course, in the third dimension. So, you know the size of a pop can, so which one's closer? You know there's not big and small ones, even though there really are minis now, but still. In terms of aerial perspectives, these are depth cues based on the understanding that's kind of inside of us. We have this implicit understanding that light's scattered by the atmosphere. Right? We know that. So more light scattered when we look through more atmosphere. So more distant objects are going to be fainter or bluer or less distinct. Right? So in these two figures, it doesn't look like there's any depth in A, but in B, you perceive it as the triangles at the top being farther away because they're kind of hazier and bluer and they have less contrast. This figure actually shows a real world scene with the cue of aerial perspective, right? So the less clear mountains seem farther away, seem to have more depth. Vanishing point is when you're looking at a scene and it's the apparent point where any parallel lines are receding that they converge at. So this is a simple demonstration of the vanishing point on the left. On the right, it's a more realistic depiction of vanishing point. So you can see that the vanishing point appears to be in the very center of the image, and all the angles on the ground and in the buildings angle toward that vanishing point. Whereas linear perspective, it says that lines that are parallel in the 3D world seem to converge in a 2D image as they go further and further away. So you can see that on the ground here. They seem to go closer and closer together. Motion parallax is when you're moving and images that are closer to you move faster across your visual field than stuff that's further away. So the brain, you know that you're moving, and the brain uses all this information to calculate distances in stuff that's in your environment that you're seeing. Head movements and any other movements between the observer and objects they're looking at, they're going to reveal these types of motion parallax cues. So the figure's showing how objects in the visual field are going to change position as you kind of travel past them on the moving train. So you're kind of sitting there and you're looking at them as you go. Oops. So when the position of you, the observer, changes, the position of the background with respect to any object that's up here in the foreground, it changes more for nearby objects like the cow or the flower, right? It changes the most. Then they change for more distant ones like the tree, right? And especially like the mountains in the very back. So we talked about accommodation before. So recall that's where the lens adjusts its size to focus stimuli on the retina very clearly. Basically, your lens gets fatter as your gaze is directed toward closer objects and thinner for farther away things. Two other things that the eyes can do are convergence and divergence. So convergence is basically crossing your eyes, like turning them inwards to focus on something that's nearer to you. So if you focus on your finger and put it closer and closer to your eyes, you're going to cross them. You're going to converge them. Divergence is the opposite. So that's the ability to turn your eyes outward to focus on farther objects. So the visual system has access to these physical cues and uses them to help you infer the distance of objects that you're fixating on. So those were monocular depth cues. Using one eye, you could just have one eye and still get all that information about your environment. But as we know, it's better to use two eyes. So these are binocular depth cues for perception. So using both eyes to perceive depth is called binocular depth perception, which is also known as stereopsis. So this arises from the fact that our eyes are about 6.5 centimeters apart on average, but more for some, less for others. So each eye has that slightly different view of the same nearby objects. So the differences in the two images, that's retinal disparity, as I mentioned before. So yeah, you'll appreciate that you have stereoscopic vision if you try and touch your two fingers together in front of you. You do that first with both eyes open and then just do it with one eye open and kind of see how good you are at doing it. Now think of like a way harder task, like much easier. It's going to be much easier if you use both eyes rather than just one eye. So there's a geometric concept that states that points on the retina where the monocular images of an object are formed are at the same distance from the fovea in each eye. Those are corresponding retinal points. So the figure on the left here, it shows the guy looking at 4 different crayons. So he's focusing on the red crayon, and the image on the right, it shows that the image of the red crayon falls on the fovea of both his eyes, right? So the right and the left, that would be the fovea of the dotted line. On the left, we see that the retinal image that he's seeing, that the blue crayon, right, is going to be at about the same point, and same with the brown, but the purple one is the one that kind of moves around, and the images are upside down because of the optics of the eye. Remember, we saw those before. So yeah, the two crayons are at corresponding retinal points, so the foveas, and the purple and brown crayons, they look like similar points as well, but they will have some retinal disparity. Yeah, and yeah, the blue crayon, I'd say, it looks like it happens to be at a corresponding retinal point in both eyes. So this tells us also that objects don't need to fall on the fovea on each eye to be in corresponding retinal points. Oh, this is just briefly, we used to have these stereoscope thingies, not like this, but a little different. It's basically you present an image to one eye and a different image to the other eye. And your brain kind of puts them together. Free fusion is a technique that people use to cross their eyes and uncross their eyes to view a stereogram without a stereoscope, like the magic eye pictures that you might have seen before. So try and do this one. I don't know if it'll work because it's on a computer. I forget what it is. I don't remember. So if you can't do this, you would be called stereo blind. Most people who are actually don't even realize that they are, because like you don't get a lot of chances to do this. So usually it's thought there's no critical period in terms of developing stereo blindness. But what it is an inability to make use of binocular disparity to see these things and process images. So it can result in from lots of things, like a childhood visual disorder, for example, like strabismus, or other things as well. Not enough, getting outside and doing fun stuff time, for example. Another thing that you might relate the stereopsis is 3D movies. So to look 3D, both eyes have to get a slightly different view of the scene, just like in real life, but a bit exaggerated. So early methods for seeing 3D movies involved the red and blue glasses, but current methods use polarized light and also polarized glasses to really ensure that each eye is seeing a slightly different image. So this gets implemented in the human brain because input from the two eyes has to converge onto the same cell, which we know definitely does happen in our visual system. And there are lots of binocular neurons that do respond best when the image is on corresponding retinal points. It's not all of them that have a preference for a certain stimuli. But of course, there are many other binocular neurons that respond best when there are similar images out there and they occupy slightly different positions on the retina. Yeah. So the figure is just showing binocular receptive fields of single disparity sensitive neurons. So yeah, you can see the fixation point, right, and which neurons are going to fire the most. So using these depth cues, stereopsis can be used as metrical and non-metrical depth cues. So some cells in your visual cortex, they're just going to code whether there's a feature that's in front or behind a plane of fixation, and that's only going to tell you non-metrical information. But there are other cells, as we know, there's tons of different cells, that these other cells code the precise different distance of whatever feature from the plane of fixation, so giving you a metrical depth cue there. So depth perception, again, results from combining lots of different cues together, right? It's a combination of the knowledge that we have, the current stimulus, and the knowledge about conditions of the world, so, what is or what is not likely to occur. And in turn, this means that prior knowledge can influence our estimates of the probability of an event. Like how likely is it that these pennies are actually a different size, right? So there's 3 different potential solutions that could lead to a penny image. And how you interpret the images of the pennies depends on the prior probability of you using pennies, and what size are they, and can they fit together like that, and what would that be like, right? So pennies are almost always the same size. There might be very tiny differences. So the situation in part A here is the most likely outcome, much less likely than situation C or B. Right? So these sorts of considerations are all taken into account by our visual system and the rest of the brain when interpreting ambiguous stimuli like this. So next question, are these 2 people the same height? Like the people, if they stood up, they'd be the same height. So actually, yes, the images of the 2 men are actually the same length, like if they were measured in their height on the screen, okay? But they look to be different sizes because of the railroad tracks, making the man in the upper image look farther away. Okay? So definitely there's kind of illusions that can be made using these depth cues. So our visual system is taking everything into account, the depth cues, but also prior knowledge and everything else when interpreting size of the object. Right? So do you see any other cues that make the person at the top look farther away? Right? So there would be relative height here, also linear perspective, and I guess a bit texture gradients using the logs and the train tracks. But all things being equal, if 2 images are the same size on the retina and one's farther away than the other, then the farther image must be bigger. That makes total sense. But in this case, they're the same, take up the same space on the computer screen. In this one, we're looking at the vertical red lines. So notice that the vertical lines on the right, they kind of look to be the same length, but on the left, they seem to be different lengths. So there's lots of cues that give rise to this type of illusion, like relative height, linear perspective, and texture gradients. Right? It's the same as the last one, but they look slightly different in length because they have different kind of backgrounds there. In terms of developing binocular vision, again, abnormal visual experience would disrupt it, and there is a critical period that it's developing. So that's the time, again, when the person is really susceptible to changes that might happen or injuries that they might get. We already talked about strabismus, but esotropia is strabismus where the one eye is inward, and exotropia is strabismus when the one eye faces outward. So in terms of these figures, the figure here on the left shows that things like stereoacuity, right, that develops into adult levels, it's thought to be quite quickly, but within the first 6-7 months of life. But if there's a problem like strabismus, then it probably doesn't develop very much at all. In terms of this figure here on the right, the idea is that that esotropic eye, the strabismus where one eye points inward, right? The idea is that your stereopsis, your binocular depth perception, it develops to adult levels basically within the first 6 to 7 months of life. But if there is something wrong, like strabismus, then it doesn't develop really at all. Like it just has a huge drop off. So definitely need proper physical functioning to develop. So have a great weekend, and I'll be back with another video next week.